I couldn't be more proud to invite Dr. Kaplan to share with you this morning. I think you all, he's a man who needs no introduction, but just our words of gratitude. So with that, I will bring up Dr. Kaplan.
Thank you, Michelle. As Michelle said last night, she used the term Thanksgiving. I'm very grateful and very thankful to be back here again. The first time I was with the families was in 1991 when Jeannie Pieper, very soon after Jeannie Pieper started the organization, the IFOPA. And we met in Florida. I think there were 8 families, and now there's many, many families and almost 1,000 patients around the world, and we're far, far along in our quest to find a successful treatment and a cure. I'm from New Jersey, although I live in Philadelphia. I grew up near exit 9. You have to identify yourself by an exit if you're from New Jersey. The title of my talk this morning is FOP, the Leaky Faucet. And I think that certainly this was mentioned before, but the scope of activity in the FOP community is simply breathtaking. And Eileen Shore, I don't know where Eileen is in the audience, my colleague for the past 30 years, Eileen Shore, maybe she's not here today. But Eileen and I can remember a time when we had to beg the pharmaceutical companies to be interested in FOP. And I think just before the gene discovery in 2006, one of the pharmaceutical companies that was interested in what we were doing came to talk to us and said, well, you're not ready for us yet. And we didn't initiate the exchange, they did, but they said, we're not ready for us yet. And things changed, and they changed rapidly, and they changed within a day. And our entire focus on the research end of this has been to understand the cause of FOP so that we could use that to establish a treatment and eventually a cure. And we heard over the past 2 days the possibility of maybe even someday, hopefully, reversing some of this problem with FOP, some of the restriction of the joint movement. Anyway, the entire landscape of the FOP community changed in one day. And it changed when the FOP gene was discovered in 2006. And on this slide, you see Mei-Chi Su, a scientist that has worked with Eileen and me, for the past 30 years. And we had the candidate gene approach, and we used a candidate gene approach. We used a positional approach to locate the FOP gene, and we eventually found the FOP gene, and it's one misspelled letter out of 6 billion letters in the human genome. One misspelled letter causes all this problem. And we published this in 2006 on April 23rd. That was the day that really changed the FOP community forever. And that was the day that we published the paper, A Recurrent Mutation in the BMP type 1 receptor causes FOP. And as I said to you before, the journal Science published an editorial the week later, and they said, really, what took you so long? But it was really a tongue-in-cheek comment, because they were saying that it really takes a community to find the answers to these things. It doesn't take one person, it doesn't take one scientist, and perhaps today we have the tools and technology to find the gene in a day. Maybe a high school student could do it, but it took us a long time, and during that time, we built up a community together, and it's that community that's here today, that's represented in the scientific community that was here over the past several days, that's now a worldwide enterprise. This went from the backwaters of medicine to the mainstream of medicine within a day. And several weeks after the gene discovery, which is the focus for FOP, the New York Times published a story in the main section of the health section of the Science Times, and they said, Mike Mason wrote this story, and he said, finally, with genetic discovery, hope. And that's the key word, hope, for escape from a prison of bone. We hope that someday there will be such effective treatment, such effective suppression of the bone development, such effective suppression of the inflammatory triggers for FOP that we'll be able to remove some of the bone and liberate some of the joints and turn what we think is a horrible, what we all know is a horrible nightmare disease into something not much more than an inconvenience. And the FOP gene is a powerful target for drug discovery. In fact, it's the bullseye of the target. And everything, it's almost a dizzying array of things that are going on now in the FOP community. And I can imagine for some of the new families, it must be extremely dizzying. Like, is this drug better than that drug? What are the clinical trials? But they all relate to the center of the bullseye. Some treatments are upstream of the gene. Some treatments are onstream of the gene. Some treatments, clinical trials rather, are downstream of the gene. Some are on the side stream of the gene. But they all relate to this genetic target of the genetic cause of FOP. And I think it's really exciting because the clinical trials are really focused on different targets. And it's possible, perhaps someday, to gather these treatments together and for any one person have a personalized approach to the targets for medicine, for FOP. So I'm going to use an analogy that you're going to see in a few minutes to explain all of the efforts in the clinical trials that pertain to treatment of FOP. On this slide, I've shown a cumulative index of the pharmaceutical and biotechnology activity and interest in FOP. And there was some before the gene discovery, but nothing tangible. And then you can see how the slope of the line has changed from the interest of the FOP and pharmaceutical community from before the FOP gene discovery to after the FOP gene discovery. Finally, there was a target to focus on that, as Regeneron said, from science to medicine, there is a target and a potential to treat this disease. And now there have been over 30 companies that have made inquiries into FOP. There are 13 different pharmaceutical companies that are developing drugs for FOP, and countless scientists from around the world that are involved in the research of FOP, all focused on the stream from the gene that causes FOP. And so soon after the discovery, my colleague Eileen Shore took this one misspelled letter and she inserted it into the genome of a mouse and got a mouse that had FOP. And this is really a remarkable discovery because it's one thing to have a genetic condition, but there's no guarantee that an animal model is going to reproduce many of the features, almost all of the features of that genetic condition for study. But this mouse did, and it has short toes, as you see there, that mimic the short toes. And it has many of the congenital features of FOP that we know in patients. The fusion, not because of heterotopic extra bone, but because the joints are misformed in the cervical spine and throughout the spine. Also the osteochondromas, the aberrant growth plates or light growth plates that form on the bones. And in addition, the mouse or these mice formed bone either spontaneously or in as a consequence of physical trauma, and it mimicked the entire spectrum of bone formation that we see in patients. So this then became not only a proof that one letter caused classical FOP that's present in 97% of individuals, 96 to 97% of individuals around the world, but also it was a great tool for the pharmaceutical industry, and there have been many iterations of this mouse model from Regeneron, from the IFOPA, et cetera, that are from the Goldhamer lab, that are used around the world now, that enable us to study the effects of different drugs, different potential drugs for FOP on the process of FOP that we can't study in humans. And so, because biopsies are anathema, so we can actually study the process. And so Aris has colonies. Aris probably has treated more mouse patients than I've treated human patients who have FOP. And many of the scientists now have access to these models to test their drugs that are upstream, downstream, sidestream of the FOP gene discovery. So if ACVR1, which is the gene that causes FOP, that one letter misspelled in that gene, imagine the genetic factory. Each of us has 2 copies of the gene, one from our mother, one from our father. And in the normal ACVR1 factory making the gene, imagine it's a faucet. Imagine the gene is a faucet. The gene is basically an instruction, a blueprint for making a protein. And imagine the protein, or imagine the product of this blueprint was a faucet. And in the normal factory, normal faucets are made, and in the FOP factory, every other faucet is normal because one copy of the gene is normal, and one copy of the gene has this one misspelled letter. So there's an abnormal faucet. And in the normal faucet, imagine the faucet is the protein, the ACVR1 protein that's encoded by the gene. And imagine the water coming out of the faucet is a signal to make more bone. In the normal faucet, the faucet should be off when bone formation is not needed, and the bone morphogenetic proteins, the BMPs, which are other proteins, turn the faucet on and cause the signal, which we know is called SMAD1, 5, or 8. These are protein signals that go into the nucleus of the cell. But basically, I'm going to use a metaphor or an analogy, as Adam said yesterday. And this is the analogy for making bone in the normal faucet. In the FOP faucet initially, we found, and others found, that the faucet is drippy. It's leaky. When it should be turned off, it's leaky. And that leakiness stimulates what we now think are the congenital features of FOP, the short malformed toe. And as we now know from research done by Will Towler at the University of Pennsylvania and also other scientists throughout the world, that the congenital features of FOP are probably, not definitely yet because it's not absolutely known, but probably caused by that drippy faucet. And they cause the fusions of the neck, the stiffness of the neck, some of the abnormalities that we know in some of the other joints that I'll show, like the hip and where the ribs join the spine. And the ribs join the spine in 3 different places. They join the spine. So there's 12 ribs on each side. So there's 36 possible joints. There's 72 joints where the ribs join the spine. And if there's any asymmetry in there, even without extra bone formation, with growth, there is regressive curvature of the spine. So we know that many joints are affected by this probable drippiness of the faucet. And we thought that the bone morphogenetic proteins turn that faucet on, exploding the faucet. It's a drippy faucet, but if you turn it on, it sort of explodes with water all over the place. And again, the water is the metaphor for making the bone. So it's like this. The FOP faucet is drip, 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 and that probably causes some of the congenital features of FOP. the exploding faucet. I don't have a picture of an exploding faucet, but imagine water splashing all over the place. That would be turned on by the BMPs. And from scientific studies done in our lab and also now throughout the world, there is ample evidence that the FOP faucet is both leaky and BMP dependent. That means that it's leaky when it should be turned off, and when it's turned on, it produces a lot more signal than it should, a lot more water than it should to make bone when it should. And part of the leaky faucet is due to this misspelled letter. This one misspelled letter in the gene causes a different amino acid, a different component of the protein to be placed in this position inside the cell, and it causes this particular region of the gene to be open rather than closed. This closed region of the protein is a binding site for a very important protein called FKBP12, and that acts as a safety lock on that faucet. And when the BMPs bind to the outside of the cell, or the handle turns on the faucet, that protein goes off, that area opens up, and the water comes out. But with FOP, that area is completely open. The FKBP12 can't bind as a safety lock and allows for the drippiness of the faucet. It's a little bit more complicated than that, but that's essentially what's going on. And that's essentially what's going on in the protein that causes the faucet to be so abnormal. And so what I've shown on this slide is what we know is the drippy faucet, which we think is the cause of the abnormalities in the joint formation. So too much of this signal can cause too much bone later in life, but too much of this signal when the embryo is forming causes the joints not to form properly. And some joints are more sensitive than others. The toe is probably one of the most sensitive joints in the body, and we all recognize that as a cardinal feature of FOP. But there are other joints affected as well, such as the joints where the vertebra joint in the back. About 50% of people with FOP have an abnormal thumb. I've also shown there are abnormalities where there is fusion of the ribs joining to the spine, and it causes inability of the chest to expand properly. The hip joint doesn't form properly. There is a dysplasia or an abnormal growth of the hip joint. We also know that there is osteochondromas of the knee. And going back to the cervical spine, we discovered that when many of you, the parents, have told us that the children don't crawl, they scoot around on their butt, and then they get up and walk. Well, you can't crawl if you can't extend your neck, and you can't extend your neck if your joints are fused. And so many of those joints are fused because of the drippy faucet that causes the joints not to be specified properly during embryogenesis. And then postnatally, the BMPs, we thought, turn on the faucet, exploding the water all over the place, sending the signal to create this extra bone. So in the old school, we thought that the bone morphogenetic proteins turn that faucet on when bone is needed to form. But then in 2015, the scientists from Regeneron, also some scientists from Japan, but Aris Economides and his group found something extremely interesting and extremely important. And they found that Activin A, which was not thought to have anything to do with any of the bone-forming faucets, actually turns the faucet off under normal conditions. The bone morphogenetic proteins turn the normal faucet on, but Activin A turns that faucet off. And we heard in the last several days that Activin A probably acts to block the bone morphogenetic proteins from binding to the normal faucet and turning it on. We don't know exactly what that means yet in evolution and in the normal functioning of our bodies, but Activin A actually acts to turn the normal switch off. And the scientists from Regeneron, Aris and his group, found that the old school thought was that an FOP was that it's a drippy faucet and the bone morphogenetic proteins turn it on. Yes, they do, and they turn it on in an explosive way, but Activin A acts in a very strange manner. Instead of turning the faucet on, it acts with the bone morphogenetic proteins to turn the faucet off. It acts with the bone morphogenetic proteins to turn the faucet on. This was an extremely interesting discovery. And Aris and his group took Activin A and a collagen sponge implant, and they implanted it in an animal, in an FOP mouse muscle, and they found that it caused bone, but again, as suspected, it didn't cause bone formation in the normal mouse. In fact, it probably did the opposite. It caused it actually blocked the bone formation in the normal mouse. And they found that antibodies to Activin A actually blocked that extra bone formation, both from a sponge in the normal mouse, but it also blocked the bone formation completely in the FOP mouse. And that was the basis for using Activin A antibody that we now know is entered into clinical trials. The clinical trials are full and completely enrolled, I should say. And I understand from my colleagues at Regeneron that in the next several months, the principal investigators from around the world will get a glimpse at the first data from the double-blind portion of that study, and all the patients now are enrolled in the second part of the study. But using this metaphor, the X there represents the antibody that prevents the Activin A, that basically holds the hand and prevents it from turning on that faucet, turning on the ACVR1 faucet, and hopefully there's less bone and less flare-ups, and we'll see. We'll see. Another very important discovery was made by my colleague Maurizio Pacifici and his colleagues from Children's Hospital of Philadelphia. And Maurizio has been interested for many, many years in growth. What causes the bones to form normally? They form, we know, in normal growth, they form by endochondral ossification, which means a cartilage model. It's sort of like the way the lost wax way of making a sculpture. So bone is not made directly. You have a cartilage model first, and then the cartilage is the scaffold on which the bone is formed. And cartilage consists of a lot of water. There's a lot of proteoglycans in there, which are bristling with negative charges, and the negative charges suck up positive charges, where positive charges are on water. Water is like, so cartilage is like a sponge. It's a great way to make bone very quickly. That's how our skeletons grow in length. That's how our bones grow in length. Our bones grow in width by intramembranous ossification, which is sort of like apple strudel, layer by layer by layer. It's also a very robust way of making bone, but a much slower way of making bone. Well, we know in FOP that the extra bone is made by endochondral ossification. It's made by this cartilage model. And Maurizio found that palovarotene, or the entire class of molecules called retinoic acid receptor gamma agonists, that's a mouthful, but palovarotene is a member of that class, blocks the transformation of the fibroproliferative stage of bone formation in FOP to cartilage bone formation. So on this cartoon, I've outlined that the muscles don't turn into bones, but the muscles begin to degenerate through inflammation, and then the muscles get destroyed, and then there's a tissue stem cell, which David Goldhamer and John Lee Shepard and their colleagues have identified over the past several years. I'll talk about that. And then that is responsible for forming those cells are responsible for forming the explosive fibroproliferative lesion, the cartilage formation of the bone. But the palovarotene blocks that step. It doesn't block, I mean, maybe it blocks a little bit of the early stages of inflammation, but it's really designed to block the fibroproliferation to the cartilage stage. And Maurizio and his colleagues tried this out in mouse models, both in the wild-type mice, BMP implants, in Dr. Machina's Q207D mouse, which is not the mutation that we see in FOP. It's just one amino acid, one building block away from the FOP mutation, but it's a very strong mutation. And then, of course, in the R206H mice. And there's a dose-response curve, so the more palovarotene that's given, the less bone there is. And we all know that there are clinical trials, phase 2, and that we have the data for, and phase 3 clinical trials that are ongoing with palovarotene, and there does seem to be a less bone there. It doesn't completely block the bone. We've learned in the phase 2 trials, but it dramatically blocks the bone, about 50 to 70%. And we'll see in the phase 3 trial how active that is. We're very concerned and cautious about the effect on the growth plate in younger children, and that's being monitored. But this is a potential trial. Now, this is a trial, not a potential trial. This is a trial. And using the same metaphor, palovarotene is sort of like an umbrella. It blocks downstream of the inflammation, and so perhaps it doesn't block the bone formation completely. Sort of like an umbrella in a hurricane. Not going to prevent you from getting wet in a hurricane, but we all carry an umbrella on a rainy day, and the palovarotene may be very effective, and it works downstream of the BMPs and Activin A and the Faucet. So it probably doesn't block the inflammatory phase of flare-ups, but it may block the bone formation downstream. And we'll see. We'll see from the studies whether or not this is true or not. We also know that the FOP gene mutation doesn't turn a normal faucet into an exploding faucet. It turns a normal faucet into a drippy faucet. And what turns that faucet on? Well, I could talk for hours and hours and hours about the work that we've done at Penn and the work done in many laboratories around the world about this, but there is increasing evidence now, almost incontrovertible evidence from many studies around the world that inflammation and hypoxia turn low oxygen tension in the cells, turn this faucet on to an exploding faucet. And this is enabled by the BMPs and especially Activin A. And there are now many clinical trials going on and others that are in preclinical development to actually block the inflammation and the low oxygen, and they're sort of two sides of the same coin, using HIF-1 alpha inhibitors, mTOR inhibitors, anti-inflammatories, immune suppressants. These are all different ways of perhaps blocking the inflammatory triggers that trigger Activin A and the bone morphogenetic proteins to open up this abnormal faucet, this damaged faucet, and cause it to explode with activity that lead to bone formation. So this is another way to perhaps tame the faucet. Over the past several years, there has been an extraordinary development in identifying the cell, or the cell type, in muscle, probably in tendon, ligament, connective tissue, fascia, aponeurosis around the muscles, not just the muscles, but also other connective tissues. The cells called fibroadipogenic progenitor cells, they are not muscle cells. They are cells in the muscle tissue, but they do not form muscle, and they normally don't form bone. They act as conductors to help in the repair process of when a muscle gets injured. And it turns out it's a very long and beautiful story from the Goldhammer lab and other laboratories, Paul Yu's lab, and other laboratories around the world. John Lee Shepard has done remarkable work in this area and shared some of that with us in the last several days. But they've identified these cells, and they've turned on the ACVR1 gene, the FOP mutation in the ACVR1 gene in just those cells, and it's enough to cause the extraordinary bone formation in FOP. And these cells are not very abundant, and they're in the muscle tissue, and they have a certain signature. So on the membrane of these cells, there are certain proteins that are expressed, like SCA1 and platelet-derived growth factor receptor alpha, Tie2, And they are not endothelial cells, they are not blood vessel cells, but they reside near blood vessel cells. And we think that these are these cells. There are probably other cells, but these are some of the cells that are responsible for the explosive bone formation. So it might eventually someday be possible to target not the liver cells and the brain cells and the spleen cells and the nose cells and the toe cells, but only the cells in the connective tissue that are responsible for forming bone. So that's why it's really important to understand this. So imagine a fibroadipogenic progenitor cell, which is going to be the cell, or certainly one of the types of cells in the muscle, in the connective tissue that causes bone. This cell has many different receptors. It does a lot of things normally. It has what we call the ACVR1 receptor, the ALK3 receptor, the ALK1 receptor, the ALK6 receptor. Well, imagine a cell as a room. Imagine it's like a bathroom. Imagine the ACVR1 receptor is like the faucet in the sink, the FOP receptor. And imagine the ALK3 faucet is sort of like the one that runs the toilet. And the ALK6 faucet is like the one that is in the shower. And the ALK1 faucet is in the one that is in the bathtub. And nature didn't design these faucets. They evolved over time, and they're very similar to each other. So you want a drug that turns off the leaky faucet, which is the one in the sink, but not the toilet. You don't want to shut down the toilet, the shower, or the bathtub. You just want to shut off the sink. Well, Paul Yu and his colleague, Ken Block, and Chaz Hong, when he was with them, you probably don't know Chaz, but he's now at University of Maryland. But Chaz discovered a molecule called dorsomorphin, and he discovered this right after the gene discovery, right after the FOP gene discovery, or actually discovered it before the gene discovery, because they were looking for a tool to turn on and off the bone morphogenetic protein pathway in other organs as well, and especially in the heart and the lung. And they had no idea that it might be applicable to FOP. Well, it turns out that the early dorsomorphin turns off the not only the toilet, not only the sink rather, the broken faucet, but the toilet, shuts down the toilet, shuts down the shower, shuts down the bathtub, shuts down all the other faucets that are similar to ALK2 or ACVR1 in that bathroom. And so it's not really conducive to use in a clinical trial. But Blueprint, Kuros, Insight, BioCryst, AstraZeneca are all making or have made molecules that are going to be highly specific, highly selective for the ALK2 receptor and for the FOP receptor. So they're designed to shut down the toilet, but not, shut down the sink, shut down the broken sink, but not the toilet, the shower, or the bathtub. And we heard about that in the past several days. And those, this is a model. The green is a model of the ACVR1 gene, and the blue is a molecule, is a kinase inhibitor. We talk about kinase inhibitors. These are small molecules, pill-like forms, that bind to the ATP binding site of the receptor and basically shut down the faucet. And so, as you see there, the circle is around the sink. We want something that's highly selective that actually blocks the faucet in the sink, but not the other faucets that look similar. And soon, these will enter clinical trials, again, using the faucet metaphor, the faucet being the protein encoded by the ACVR1, by the FOP gene, and these are like little plugs that will fit in the faucet and block the water from coming out of the faucet after birth and stop the exploding water from causing hopefully a flare-up and the extra bone formation. And so these will now enter clinical trials in the next year, some in the next more in the next several months, probably some within a year, some within 2 years within the FOP community. We believe that although 3% of patients have FOP variants, and some of you are actually in this room, some are more severe, some are less severe, these are mutations in the same gene, but different letters substitutes, different abnormalities that cause this faucet to be either less leaky or more leaky than the classic FOP faucet. But we believe that the kinase inhibitors, these plugs that screw into the faucet that actually will hopefully inhibit the inflammation and the bone formation, will be applicable to FOP variants as well as classic FOP. And certain trials, for example, the anti-Activin A trial and also directly blocking the faucet itself will probably be will certainly be applicable to both the classic and the variant forms of FOP. And so what I've listed here is all the different ways that we can block that FOP faucet, or the protein ACVR1, the mutant protein damaged by one letter caused by the mutation in that one letter in the FOP gene. And so we have Activin A antibody. There is one pharmaceutical company, Regeneron, that's working on that. And that blocks the antibody. There are 2 pharmaceutical companies that are working to develop antibodies to the receptor itself, not the hand that turns on the faucet, but the faucet itself. There are 5 pharmaceutical companies that are working, private companies that are working with academic partners to develop, and we'll hear more about that this morning, to develop signal transduction inhibitors that block the faucet, that screw into the faucet. There are 4 possibilities for that are being explored to block the inflammatory triggers of that faucet, and one that blocks downstream. So I just wanted to give you a sense of how each of these clinical trials, each of these ongoing or potential clinical trials, are working on this pathway. They're blocking different targets, and each of them will have its own series of efficacy, potentially, and each having its own possibilities for side effects as well. So, but these are different targets. It's very important not to go away from here and think, well, what's a better drug? Because FOP is a complex disease, and for each individual, depending whether it's a child or an adult that already has some of the complications of FOP, we may use different medications. It would be wonderful to have different medications in our medical toolkit that we could use to quiet down the flare-ups of FOP, quiet down FOP. We don't know if we'll be able to use these drugs for decades at a time. Certainly when we treat the mice, it's for weeks at a time. But in humans, it's going to be for decades at a time. Is a person going to be able to tolerate these medicines for a decade at a time? Maybe we need a holiday from these medicines. So just like in asthma or diabetes or in hypertension or any other common disease, when we go to the doctor, and it's very nice to have, for the doctor to have several medicines in the toolkit and say, try this medicine. We'll see if this medicine works. This is not as effective in you as I was hoping. Let's add this other medicine, or let's try this medicine instead. Or for this stage of the disease, I think this medicine is better than that medicine. So it will be really, really good to have many medicines in this toolkit because they're hitting many different targets. We're going to probably have to try each one individually, but before we try them together. And there are many other, we heard in the last several days, that there are many other ways to potentially block this pathway. There is a possibility of genetically blocking this pathway, removing the faulty faucet in the abnormal cells, or not using the sink, or using the normal faucet but not using the abnormal faucet in the sink, in in these cells. But that's for the future. Those are things, and we now have the tools to investigate those, but those are the things that are going to be for the future. So right at the center of the bullseye here, and I've put a box around that, is ACVR1, or the R206H mutation. I've listed the different targets from soft tissue injury to spontaneous flare-ups, all the way down to the formation of heterotopic bone. And I've listed potential treatments on the left-hand side. Many of these, or all of these, are being investigated. All of them are certainly being investigated by researchers around the world. Some have entered clinical trials, and some will enter clinical trials. But these are various ways of blocking the targets. Another analogy would be, imagine a terrorist. You want to get a terrorist, but you want to take them out. But before you take them out, you have to know where that terrorist lives. Are you going to use a pistol? Are you going to use a shotgun? Are you going to use a rifle? Are you going to use a cruise missile? How are you going to take this terrorist out? And it's very, very important to have many, many different, to know what the terrorist is. We know what the terrorist is. It's the ACVR1 mutation that encodes this protein. But there are downstream, there are many, many different targets for this pathway. And I hope I've given you some understanding of this. Now, several months ago, when one of our patients unfortunately was deceased and left her skeleton to the Mutter Museum of Philadelphia, of the College of Physicians of Philadelphia, the Philadelphia Inquirer did a big story on her gift to the museum, and also on, as a sidebar, drug therapies that were in sight for a bone-forming disease. And I highlight what they wrote there in yellow, and they said, how a 2006 gene discovery made an obscure condition a worldwide enterprise. And it certainly is a worldwide enterprise. And now our ultimate goal is to have a treatment and a cure for FOP, and maybe even someday reverse the findings of FOP. But on the positive side, the green check, we want to stop flare-ups. We want to prevent flare-ups. We want to stop the bone formation that's not due to flare-ups. We don't exactly know what that is yet, but we're learning that, and with the animal models, we will learn that, and hopefully in the near future. We want to essentially maintain function. That's the essential goal, is to maintain function. Now, most of the clinical trials are only going to go on for a year or two at most, 3 at very most, but probably a year or so. And so the changes, as Dr. Kakis has said in the last few days at the Drug Development Forum, and as we know from some of the clinical studies, the functional decrements from this bone formation are not necessarily visible within the time frame of a clinical trial. And so we're using the surrogate measures of how much bone is formed. We all know that a lot of bone forms in certain areas, like the back, around the hip, around other areas of the body, but the elbow, maybe a smaller amount of bone forms, and if it forms in the wrong place at the wrong time, it may actually completely lock up a joint, whereas you can get a massive amount of bone around the thigh that affects the knee and affects the hip, but doesn't lock either joint up. So we know that bone is not the amount of bone that forms is not a complete surrogate for the functional decrement that's going to occur, but over a long time, or over a short time, it's probably an indicator of what's going on with the disease and whether that drug will have an effect over a long period of time. And if any of these drugs are approved, hopefully they'll all be approved, but we'll see. If any of these drugs are approved, there's going to be a long-term follow-up to see how the patients are doing on these drugs over long periods of time and seeing if there's a functional decrement. And we want to slow the joint degeneration. That's a really interesting question, because we know that comes, or we think that comes from the drippy faucet. And so the question is that a fait accompli sort of at birth, that these joints are going to degenerate over time and cause an arthritic condition that progresses through life, even if the extra bone doesn't form? Or will some of these medicines maybe stop or even reverse some of the joint degeneration that occurs? We also know that many of these drugs have side effects, or almost all drugs have side effects. I mean, take a glass of water, the water goes down the wrong pipe, you can choke on it. So even water, which sustains life, can cause side effects if you don't take it properly. So of course drugs are going to cause side effects. So we have to look at the side effects in individuals. We have to look at off-target effects, allergies, non-response. Maybe there'll be some rebound if a patient is taken off the drug. Maybe they'll have resistance. Are they going to take the drug? Are they tolerating the drug? What's the access to the drug? And finally, what's the drug going to cost? So all these things have to be taken into consideration, and many of them have to be, and so that's why we need multiple drugs to treat FOP, probably not just one. As Adam Migris, who's in the audience today, wrote to me as a little boy when he came to see me in Philadelphia, he said, "Thank you for seeing me at the hospital. I had fun on the tour. I liked the ice cream. I didn't like the needle. Hope to see you again. Please help fix my FOP." And I think that's the bottom line. And again, it's hope. What all this really amounts to is one word, and that's hope. And hope is a very powerful medicine. Very, very powerful medicine. And that's what that's why we're all here today. We have hope for the future. We have a vision for the future. It's not hopeless like it was 30 years ago. We didn't have a target. Now we have a target. As Ian Kelly said, with the gene discovery, we went from hopeless to hopeful. And as Jacques Gamboa said, one of my patients from Mexico, "Dr. Kaplan, we need hope, and if we have hope, we'll be okay." This is a picture of our team at the University of Pennsylvania. We're just one of 30-some teams around the world that's working to combat FOP because we have the tools now, not just the tools that have been made available throughout the world, but the specific tools and the target. We know where the terrorist is. We know the house he's in. We know the room he's in. And we know we're designing the weapons to go get that terrorist and eliminate him. Last year in the fall, my colleagues and I wrote the 27th annual report of the FOP Research Project. When this started as the first annual report, it was just from the University of Pennsylvania. Now it's what's going on around the world about FOP. And hopefully in the new year, we'll start writing the next year's annual report, which will be available again through the IFOPA, thankfully, in the next year. And we'll see all of the different changes. We'll try to make it completely understandable. I also want to put a pitch in for the International Clinical Council on FOP, just in concluding. The International Clinical Council on FOP is 21 members from 14 countries, 6 continents. They're physicians, dentists from around the world who have the clinical expertise in caring for FOP patients, and we assembled the what you know as the FOP clinical guidelines, or the medical management of FOP. This is more symptomatic management. It outlines the basic science of FOP, the drugs that we use to symptomatically, in the old world, treat FOP until some of these newer medicines come along, and also some of the complications and precautions that are necessary in the FOP community. And at the back of this book, a booklet which is available on the FOP website and the ICC website, are the clinicians from around the world who can be contacted to inquire about FOP. I just want to acknowledge, this is just from the Penn site, I just want to acknowledge all our contributors over the years, and especially the IFOPA for its continued and wonderful support for our center, which is not just basic science research, but clinical research and collaboration to help make this a better world for all of us. So thank you.